Bismillahirrahmanirrahim. I am Haseeb Ullah from MLT 5th semester, Self-Finance at Kohat University of Science and Technology, Kohat. Today my presentation topic is the classification of biochemical tests and bacteriology lab. The discussing agenda, we will discuss, firstly we will introduce the biochemical test and their classification. Catalase test, we will discuss catalase test, coagulase test, oxidase, urease, nitrate reduction test, carbohydrate fermentation test. At last we will discuss summary of the all test. Plus, introduction to the biochemical test. Biochemical tests are used to identify bacterial species by differentiating them on the basis of biochemical activities. Biochemical activities like nutrient utilization, carbohydrate utilization, enzyme production, etc. Firstly, we will discuss the catalase test, its procedure. Mm, uh, we will show you the animated picture of negative catalase test and the positive. Catalase test is used to identify those bacteria that produce the catalase enzymes or not. Catalase is an enzyme that converts hydrogen peroxide H2O2 into water and oxygen gas. In this test, add hydrogen peroxide to a bacterial colony on a slide or in a test tube. If the bacterium produces catalase, the hydrogen peroxide will be rapidly broken down into water and oxygen gas, resulting in the production of bubbles. If bubbles appears, then it means catalase is positive. If bubbles didn't show a reaction, it means the catalase is negative. We can done this test by smear like slide and test tube. Its procedure is so simple, not too complicated. Catalase enzyme that convert hydrogen peroxide to water and oxygen gas. The test is easy to perform. Bacteria are simply mixed with H2O2. If bubble appears due to the production of oxygen gas, the bacteria are catalase positive. If no bubble appears, bacteria are catalase negative. We can show in this picture. Actually, this test is the confirmation of our differentiation of Staphylococcus or Streptococcus. It simply it show us the species or Staphylococcus or Streptococcus. We can see this is a catalase positive. Catalase positive show that Staphylococcus are present here. Streptococcus, if negative appear, it is a Streptococcus. We can see in a glass tube catalase is positive, catalase negative. There is no bubble appearance, it means that catalase is negative. If bubble appears, it means that the catalase is positive. The next one is the oxidase test. Oxidase test is a common biochemical test to determine if a bacterium produces certain enzyme called cytochrome oxidases. The test is based on the principle that cytochrome C oxidase is involved in the electron transport chain, etc. The function of oxidase enzyme is that they accept electrons from external material. Suppose this external material is tetramethyl TMPD. When phenylenediamine come in contact with oxidase enzyme, they gain an electron from it and appear a dark color. If it take or gain oxidase enzyme, if the oxidase enzyme give when oxidase enzyme gain electron, then it appear a black color. Procedure is so simple. A small amount of a bacterial colony is placed on a test strip or filter paper that has been impregnated with an oxidase reagent such as TMPD. The reagent reacts with the oxidase enzyme, causing a color change on a strip or paper. Positive result is indicated by the appearance of blue or purple within 10 to 30 seconds. If it extend the duration, so the result may be negative. First, we can see the oxidase negative. There is no color appearance in this slide. It means that oxidase is negative. If color appears between 10 to 30 seconds, it means that oxidase is positive. Next one is a coagulase test. Coagulase Staphylococcus aureus is known to produce coagulase, which can clot plasma into gel and tube or agglutinated cocci in slide. The test is useful in differentiating Staphylo aureus from other coagulase-negative staphylococci. Most strains of Staphylo aureus produce two types of coagulase, free coagulase and bond coagulase. While free coagulase is an enzyme that is secreted extracellularly, bond coagulase is a cell wall-associated protein. Free coagulase is detected in tube coagulase test, and bond coagulase is detected in slide coagulase test. Slide coagulase test may be used to screen isolates of Staphylo aureus, and tube coagulase may be used for confirmation of Staphylo. So the next one is the coagulase test principle. On which principle it is based? The bond coagulase is also known as clumping factor. It cross-link the alpha and beta chain of fibrinogen and plasma to form fibrin clot. The deposit on the bacterial cell wall is a result. Individual co-constrict to each other and clumping is observed. Procedure is First we take test and the second control on a same slide. Dense suspension of test are made on slide. With the help of wire loop, we aid the plasma and mixed with the reagents. Third one is you can see on the third slide, indicating Staphylo aureus. You can see the agglutination. You can see agglutination here. This is the coagulase test is positive. It shows the Staphylo aureus, and the control is still negative. You can see in the result interpretation if you see any type of agglutination or clot here, no, so it's positive. Staphylococcus aureus are here present. On smooth suspension, you can see the Staphylococcus epidermis. Urease test. The urease tests are used to determine if a bacterium can produce the enzyme urease, which break down urea into ammonia and carbon dioxide. A positive test is indicated by the presence of ammonia, which raises the pH of medium. You test the ability of an organism to produce an enzyme urease. Hydrolyzes urea to ammonia and carbon dioxide. The broth contains the pH indicator phenol red. Phenol red turn yellow in an acidic environment and pink in an alkaline environment. Urea is degraded and ammonia is produced. In alkaline environment, it is created and the media turns pink. For example, to urease positive, Proteo species and Helicobacter pylori, H. pylori. Urease negative, which show the yellow color. If it appear a pink color, no, so it's positive. Nitrate reduction test. It is used to differentiate the various gram-negative rods, and also mycobacteria. Nitrate reduction test principles are here following. Some anaerobic bacteria use nitrates in an anaerobic environment. As a result, they produce nitrate and nitrogen gas. Nitrite change the color of media, while nitrogen gas can be seen in the form of bubbles. We can see the nitrogen gas in the form of bubbles. Nitrate reduction test. We need peptone, agar, potassium nitrate, and distilled water. First, we need the nitrate agar. For the nitrate reduction test, we need some reagents. First one is nitrate agar, sulfonilic acid, alpha naphthylamine. How to perform nitrate reduction test? First of all, inoculate the media by sticking the slant and stabbing it to the butt with test organism culture. Incubator at 35 centigrade for 4 hours. Add 1 drop of sulfonilic acid and 1 drop of naphthylamine to the slant. Now for a change in color, if red color is seen, the result is positive. Otherwise, result is negative. After incubation of 4 hours, add 1 drop of sulfonilic acid and 1 drop of naphthylamine. If a color is red appear on medium, it will be considered nitrate reduction test is positive. Used to identify the anaerobic gram-negative rod, all entire bacteria reduce nitrate. So it is positive. It is positive. Originosa and E. coli. Negative. Here is a negative. It is the unconjugated red color after addition of sulfonilic acid and alpha naphthylamine. Here is a positive, and here is also positive. Here is no color, so it's mean it's negative. Carbohydrate fermentation. Carbohydrate fermentation. In carbohydrate fermentation is the glucose, lactose, and sucrose. These three tests are performed. This test is used to determine the ability of an organism to ferment various simple carbohydrate sugar. A small Durham tube is inverted and placed in each larger tube of liquid medium. The inverted tube can trap any gas produced. The indicator phenol red will turn yellow below a pH of 6.8 to 9, showing fermentation. In darker pinkish red above 7.4, showing no fermentation. Acid yellow, positive for metabolizing sugar. A color change has happened. Acid gas yellow plus gas bubbles. If yellow color appears and the gas bubbles appears, no, so it's mean that positive for metabolizing sugar and gas production positive. Color change plus bubbles. Negative, no color change. In a negative situation, if the carbohydrate fermentation test is negative, there will be no change in color. You can see negative carbohydrate fermentation. There is no color change, and there is also no bubbles in this tube. Positive carbohydrate fermentation, it changed the color. Here is the gas bubbles. You can maybe see. No, this is not zooming in out. Unconjugated, uninnoculated. This is a control. This is a control. Uninnoculated mean there is no sticking here. Is the negative because it didn't change the color, and no bubbles, no gas bubbles are appear. Positive, it just changes color, and here you can see bubbles, maybe small dots. If you Okay, this is the summary. So these tests are just a few examples of many biochemical tests used in bacteriology laboratory to differentiate between different types of bacteria based on their metabolic characteristics. These are the reference.